Okay. Now about the seed. What's a seed? How seed is formed? Seed is formed from the ovule. When it is formed, seed is formed from the ovule only after fertilization. Only after fertilization. So which are the structures present in the seed? Seed has seed coat, seed coat, cotyledons, embryo with plumule, radicle, then endosperm. So like that, these are the contents present in the seed. So what is seed? Seed is a fertilized ovule. Fertilized ovule is called seed. Now, structure of dicot seed. So before you go into dicot seed, you must know about endospermous seeds, non-endospermous seeds. Seeds with endosperm are called endospermous seeds. Example: maize, wheat, paddy, castor, etc. So there and all you get endosperm. Seeds with endosperm, endospermous seeds are albuminous seeds. Seeds without endosperm are called exalbuminous seeds or non-endospermous seeds. Now we are going to see about the structure of dicot seed. Cyser originum. What is cyser originum? Cyser originum. Now, actually, normally, what happens here? It's a pod. Fruit is a pod. Pod in the sense it develops from monocopulary, unilocular, superior ovary with only one locule with marginal placentation. This is what ovule. The ovule attached to the wall of the ovary by a placenta. So like that, the seed is attached to the wall of the ovary by a structure called funiculus. Funiculus. It gets detached. See, when the seed separated from that, it gets detached. It leaves a structure called scar called hilum. So close to the hilum, what will be present? Close to the hilum, micropyle is present. Micropyle is also called opening of the seed. Actually, the ovule has an integument has a wide gap at the top called micropyle. The opening of the seed is also called micropyle. The seed is protected by two layered seed coats. Seed is protected by a seed coat. Outer seed coat called testa. Inner seed coat is called tegmen. The testa, tegmen fused here. Not they are just fused. Testa, tegmen, brown colored. That's what testa. Now below that you get tegmen. Now within the seed coat there are two cotyledons. And then with a central embryo. Two cotyledons laterally and central embryo. Now the two cotyledons store food material for the developing embryo. So the embryo is a small embryonal axis. This is what called embryonal axis. The embryonal axis has plumule, radicle. First formed shoot called plumule. First formed root is called radicle. Actually, rudimentary shoot called plumule. Rudimentary root called radicle. And then part of the region between the plumule and cotyledon, cotyledon and plumule, it is called epicotyle. The part of the embryonal axis below the cotyledon and the radicle, it is called hypocotyle. So this is what structure of cyser originum, dicot seed. Now next about monocot seed. Here in the monocot seed, fruit is one-seeded. So last year we have said about caryopsis. Fruit is one-seeded, and then the fruit is covered by brown colored husk called glumes. How many glumes? 2 rows of glumes are there. One row Another row, 2 rows of glooms are present. So below that, next to the gloom, what will be present? Seed coat is present. Seed coat is fused with the seed. Seed coat is fused with the seed. Brown colored seed coat fused with the seed. Cannot be separated. Okay. Now here in the paddy seed, this is what caryopsis is. It's a paddy seed. This is an endless of fruit. Paddy seed or paddy fruit, commonly called caryopsis. Now the fruit is covered by a layer, seed coat, and then seed coat and seed closely attached. Now major portion of the seed is covered, is having the endosperm. The endosperm is covered by alluron layer. Last year we studied about it. The endosperm is separated from at one side of the endosperm where there is a small embryo. The embryo is separated from the endosperm by epithelium. Then the embryo is protected by a shield-shaped cotyledon. This is what shield-shaped cotyledon. The embryo has shoot apex, commonly called plumule, which is protected by a layer called coleoptile. And then opposite to that, embryo has a radicle. And the radicle is covered by root cap. Next to root cap, coleorhiza is present. Root cap is protected by coleorhiza. This is what the structure of paddy seed. Now about the structure of dicot seed. This is one structure of monocot seed. Monocot seed. So what is this seed? Seed is formed only after fertilization. From which one? Fertilized ovule develops into seed. Fertilized ovule develops into seed. Now you have just seen about the seed formation and all that. Now about another one, amphimixis. Reproduction involving fertilization. That is fusion between the male and female nuclei. That's what called amphimixis. Even in zoology you will study about that. Amphimixis. Fusion between the male and female gametes is called female nuclei. It's called amphimixis. Reproduction involving fertilization in flowering plants is called amphimixis. Apomixis, it's another term, another kind of reproduction, alternate to sexual reproduction. That's what apomixis. Reproduction does not involve the union of male and female gametes. Reproduction does not involve fusion of male and female gametes. It's called apomixis. Apomixis in the sense, normally reproduction takes place, sexual reproduction takes place by the union of male and female gametes. It is called amphimixis. Then sometimes what happens here, embryo is formed even without the act of fertilization. Which are absent? Meiosis, syngamy are absent in that. Meiosis and syngamy. Meiosis, haploid gametes are formed. Syngamy, fusion of the bone. All that absent in that. So the term apomixis was introduced by Winkler in 1909. 1908. It does not involve meiosis and syngamy. Which one? Apomixis. So apomixis is of two types. Professor Maheshwari described apomixis is of two types. Professor Maheshwari classified apomixis as two types: recurrent apomixis, non-recurrent apomixis. Recurrent apomixis in the sense the plants formed through apomixis can able to produce young one, whereas non-recurrent in the sense cannot able to produce young one. That's what a recurrent, non-recurrent. So recurrent apomixis you get vegetative reproduction. So already you studied about vegetative reproduction last year, this year also. Vegetative reproduction takes place through, which are the structures? Bulbils, Fritillaria, bulbs. Example, Allium cepa, onion. Runner, Mentha piperata. What is Mentha piperata? Mentha, mint. Pudina. Sakar, chrysanthemum, samandi. Now, next one about agamosphere. So, in agamosphere is a process by which embryos formed by eliminating meiosis and syngamy. Agamosphere is a process by which embryos are formed by eliminating meiosis and syngamy. How? Meiosis, without meiosis, syngamy, embryo is formed. So there are three types of agamosphere. One, adventive embryos. Adventive embryos in the sense, this is what structure of ovule. Here you get nucellus, then integument. So nucellus, integument, diploid one. Nucellus, integument, diploid one. Cells develop into embryo. So the embryos develop from nucellus and integument. Nucellus and integument. Such embryos are called vegetative embryos, adventive embryos. See, formation of embryos from diploid sporophytic cells, nucellus and integument. What happens here? Normally embryo sac is formed from megaspore mother cell after meiosis. But megaspore mother cell is formed. So megaspore mother cell is formed, then undergoes meiosis. But what happens here? Such cells will not function. But whereas one of the cell or some cells from the nucellus or some cells from the integument directly function as embryo, diploid embryo. So this kind of development is called adventive embryos. So example, citrus. Citrus in the sense, citrus and mangifera. Citrus, lemon, mangifera, mango. Diplospory. So diplospory in the sense, normally one megaspore mother cell, nucellus from nucellus, one megaspore mother cell. One megaspore mother cell develops. This megaspore mother cell divides by meiosis. Divides by meiosis to form 4 megaspores. So in that 4 megaspores, what happens here in the 4 megaspores? 4 megaspores, 1 megaspore become functional, 3 megaspores degenerate. But here, what a megaspore mother cell functions as embryo. So this kind of, but this will not take place. Megaspore mother cell directly function as embryo. That kind of structure development is called diplospory. Example, eupatorium and airworm. Next one, apospory. So apospory here, megaspore mother cell undergoes meiosis, 4 megaspores degenerate. 4 megaspore here, megaspore mother cell in diplospory, 1 megaspore mother cell functions as embryo, develops into embryo. Whereas here 4 megaspores are formed, all the 4 megaspores degenerate. So one of the cell from the nucellus or some other structure develops into embryo. That's what called apospory. Example, hieracium and parthenium. Okay, I just completed everything. Only 2 subdivisions are there. Parthenocarpy, one more polyembryonic. Only 2 sub-lessons are there. Tomorrow I will be completing everything. Then Monday, get ready for the test. I will be giving you a test. You prepare all that. Okay.